This video is a continuation of the previous video on macromolecules where we talked about carbohydrates and lipids. So we're going to finish today talking about proteins. Proteins have a diverse structure, meaning they have a wide range of functions. As catalysts, called enzymes, they speed up the rate of reaction in organisms. They get used repeatedly as they're not used up in the reaction. This is probably the most important function of proteins. Secondarily, they are also used for structural support, cellular communications, movement, and defense against foreign substances. Proteins are super important molecules, as we'll see. Amino acids have a central carbon that has a carboxyl group, an amino group, a hydrogen, and an R group attached. Amino acids differ in their properties based on different R groups or side chains. So here's an example of an amino acid. And so you can see the amino group here in orange, and the carboxyl group here in yellow, and then the single hydrogen. There's the central carbon in hot pink, and then the R group. So the R group differs for every single amino acid. So here are some of the 20 amino acids. You don't have to remember every single amino acid, but I do want you to understand the different categories of amino acids. So the first category are non-polar amino acids. So notice the different functional groups. So they're shown here. So I'm going to highlight a couple of those. So a hydrogen, a methyl group, these different chains of hydrocarbons. This has a benzene. This has like a double ring carbon structure. And what they all have in common is that they are all non-polar. So the end form of it, not looking at the other part, so we're not looking at here, because this part of every amino acid is polar. But we're looking at the R groups, so these other chains, non-polar. Okay. So then the next one we get to is polar. So looking at polar R groups, we see this one contains a hydroxyl group, this one contains a hydroxyl group, this one contains a sulfhydryl, hydroxyl, amino, amino. So these are the polar . Okay, next we have two electrically charged forms of amino acids. So their R groups contain either something that makes them acidic or basic. So the first two we have are aspartic acid and glutamic acid, also known as aspartame or glutamine. And so aspartic acid is an acid because it has a carboxyl group, and glutamic acid is acidic because it has a carboxyl group. So they donate hydrogens into the solution. And then we have the basic ones. So these basic ones, lysine, arginine, and histidine, are basic because they donate OH into solution, and they do that by accepting hydrogen. So they all have forms of amino or a nitrogen that can accept and become positive. So they're accepting hydrogens, which makes for more hydroxides in solution. Okay, polypeptides. So polypeptides are polymers built from their monomers, amino acids. So polypeptides are built. Each amino acid is joined by another amino acid by what's called a peptide bond. So peptide bonds are the covalent bond between the carboxyl group of one amino acid and the amino group of the next amino acid. Polypeptides can range in length up to thousands of monomers long. Each polypeptide has a unique sequence of amino acid. A protein is made of one or more polypeptides in a unique three-dimensional shape. So here's an example of the reaction. So remember, this reaction is called dehydration synthesis, and it's called dehydration synthesis again because notice that water is being taken out of the reaction in order to form this bond. And so this is the peptide bond that forms. You'll see I'm just going to highlight it here. So when you see a C and an N together, that's a peptide bond. So this here is a peptide bond that already was formed. As you can see, it existed before. So what we describe as this is the backbone of the amino acid, the nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen as it goes along, many thousands of polypeptides long. And then both ends of the polypeptide will have their own ending. So one will be the amino terminus. So here, the amino terminus, and the other end will be the carboxyl terminus. So we have the C terminus and the N terminus. Okay, a functional protein consists of one or more polypeptides twisted, folded, and coiled into a unique shape. So here are two examples or diagrams of what they look like. So instead of a single long chain, they're coiled and formed into some three-dimensional shape. So you can see a ribbon model of lysozyme and a space-filling model of lysozyme. Lysozyme, you may not know, is actually a material that's found in your tears, and it helps to reduce the amount of infections that your eyes get. So it actually has a job in killing microorganisms that might enter into your eyes. So structure dictates function. This is key. The sequence of amino acid determines the protein's 3D structure, and a protein's structure determines its function. A protein cannot function unless it has proper structure, proper 3D shape. So there are 4 levels of structure. The levels of structure start with the primary structure. So this is a unique sequence of amino acids. You can think of this like a series of beads along a chain as a beautiful necklace. So if you think of these, each bead is slightly different from the one before it and the one that's after it. And that sequence of amino acids of the 20 different types of beads or 20 different amino acids gives each polypeptide its own unique structure. The secondary structure are coils and folds in the polypeptide chain. So this is where the folds are regular folds or regular coils back and forth within the polypeptide chain. The tertiary structure is determined by interactions between side chains, and the quaternary structure is a protein that consists of multiple polypeptide chains. So not all final proteins have quaternary structure, because not all proteins are formed from multiple polypeptides. So here's the primary structure. The sequence of amino acids is like the order of letters in a long word. Primary structure is determined by inherited genetic information. This information is directly written on the DNA within the nucleus. So you see the amino end, you see each amino acid subunit gives the primary structure. Then this primary structure can have any change in primary structure can affect a protein's ability to function if it affects its shape. So for example, sickle cell anemia is an inherited blood disorder that's caused by a single amino acid change in the protein hemoglobin. So a single amino acid is swapped out for another one, and it causes a major disorder that we'll look at in further depth. So there's the amino acid structure at the top, the primary structure. And so it should be valine, histidine, leucine, threonine, proline, glutamine, glutamine. But as you can see, instead in sickle cell hemoglobin, the 6th amino acid in the chain is substituted from glutamine to valine. And that tiny change causes a change in the secondary and tertiary structures. So it changes the actual shape of the final protein. And this means when we get to quaternary structure, instead of a normal hemoglobin, you have a misshapen hemoglobin. This misshapen hemoglobin means that the molecules do not form the correct structure, and in the final product you see the normal red blood cell at the bottom, and then you see a sickle cell red blood cell. And this means that the red blood cells that are sickle cell shaped are more delicate and more prone to breakage, and this is what leads to sickle cell anemia. So there's just a diagram again of normal red blood cell versus a sickle cell red blood cell, and that's all caused from the change of one single amino acid. Secondary structure, there's 2 different types of secondary structure, alpha helix. Alpha helix are coiled regions, so they are things that spin around and have a regular structure. The second type of secondary structure are beta-pleated sheets. So beta-pleated sheets are folded sections that go back and forth, looking like a paper accordion. Coils and folds of secondary structure result from the hydrogen bonds between repeating parts of the polypeptide backbone. So here is a diagram of secondary structure. So you see the pleated sheet. And so you can see that there's an up and down motion back and forth, back and forth, back and forth. And it's these hydrogen bonds here that allow for that back and forth motion. And that's regular. On the other hand, we have alpha helices. Alpha helices, you have a spiral structure, and that is due to, again, the regular interactions. Okay, so tertiary structure. Tertiary structure are interactions among side chains, including hydrogen bonds, hydrophobic interactions, and strong covalent bonds called disulfide bridges. So tertiary structure, for example, you can have an ionic bond. In an ionic bond, the side chain from one amino acid and the side chain from another amino acid actually have the movement of a hydrogen atom across, leaving one positive and another negative, and creating a strong amount of attraction. Also, you can have polar forces, so you can have regions where you have that want to come together and are hydrophilic, and you can have hydrophobic areas that want to stay from water. So this area would find itself at the inside of a polypeptide. It would want to be away from water as much as possible, whereas these blue regions would want to be closer to water and would be found on the outside of the polypeptide. And then the last type of tertiary structure is a covalent bond called a disulfide bridge. So this occurs only between molecules that have the amino acid that contains a sulfhydryl group, and you may recall the sulfhydryl group are proline. And then quaternary structure is the last type. So quaternary structure is when two or more polypeptide chains form one macromolecule. So example, collagen consists of 3 polypeptides that are intertwined into a longer chain, and hemoglobin consists of 4 polypeptides, 2 alpha and 2 beta chains. So they're actually just 2 different types of polypeptides that form one molecule made up of 4 different polypeptides for hemoglobin, as we've already seen in the example of sickle cell anemia. So there's an example of the polypeptide chains in collagen, and then there's an example of the chains in hemoglobin. So you can see the 2 alpha and the 2 beta chains forming together, and you can see the iron, or the heme group, in the center, and that is actually what gives hemoglobin its distinctive color, which is red, and that is what gives red blood cells their distinctive color, which is what makes our blood red. Determining protein structure. So physical and chemical conditions can affect the structure of proteins. Alterations in pH, salt concentration, and temperature can cause a protein to unravel. Denaturation is the loss of a protein's proper 3D structure. And as we said, structure enables function. So without the proper structure, there is no proper function. So a denatured protein is unable to function. In order for a protein to actually be formed into the proper shape, instead of just being a polypeptide chain, it does require some assistance inside the cell, generally inside the endoplasmic reticulum. There are these molecules called chaperonins that actually allow proteins to enter into this sort of hollow cylinder where they have a sort of easier time folding into the proper 3D shape. So once they fold into the proper 3D shape, that allows them to come out and be fully functional proteins. Okay, this is the end of structure and function of macromolecules.